All right, to start off our immunity topic, we're going to have a look at some of the different types of pathogens, specifically focusing in really on bacteria and viruses. So some key terms that we need to be familiar with. I'm not going to go through all of these now, but the terms that you really need to focus on and make sure you're incorporating into your responses are firstly, an understanding of what a disease is, okay? Secondly, differentiating that between an infectious disease and a non-infectious disease, so one that can be passed from person to person in the case of infectious. And finally, the key word, if anything, and the title of today's lesson is pathogen. So a pathogen really simply is a disease-causing microorganism. So bacteria. We spoke about them a lot in Year 8 Science, and now we're diving into them in a little bit more detail. We use bacteria. Okay, bacteria can be good, bacteria can be bad. So some instances where we actually use bacteria, obviously in our own digestion within the small intestine. However, we can also use them in yogurt and cheese manufacturing. However, there's a really small number of bacteria which can cause disease. Now, bacteria themselves are microscopic prokaryotes. What does this mean? It's effectively stating that they have no membrane-bound organelles. So what you'll notice over on the side, or the left-hand side here, is I have an example of my bacteria, and on the right-hand side I have an example of a human cell, what we know as a eukaryote. In terms of the structure of bacteria, there are some key things that you need to know. So, number one, starting top left, some bacteria have a slime layer. Okay, not all bacteria, and this is just a very simple outer layer. Okay. Secondly, bacteria have a cell wall. Okay, this is important. Okay, it provides protection to the bacteria, and it is made up of a protein-carbohydrate combo. Number three, DNA, also very important. There is no nucleus within the DNA. However, if you've seen the recombinant DNA technology lesson, you'll know that some bacteria don't just have DNA which is floating around within their cell. They can also have a circular form of DNA which we refer to as a plasmid. Third, cell membrane. The cell membrane is not the same as the cell wall. Okay, the cell membrane here works very similarly to all other cell membranes. This is a flashback to Year 11 where we've looked at the phospholipid bilayer, no different here. Fifth, we're onto the flagella. The flagella are these tails that stick out from the bacteria, and they're simply used in movement. Cytoplasm, seen it before, effectively this fluid-like component within the cell that can appear quite granular, and that's due to the appearance of ribosomes in this case. And finally, we have a capsule. Now, a capsule, again, is there for protection. Okay, this one is made up of largely carbohydrates compared to the cell wall, similarly used for protection, but that's made up of a combination of protein and carbohydrate. So, what are some of the structural characteristics that you should know? Well, number one, they're microscopic. We can't see them with the naked eye, and they are unicellular. Their entire being is made up of one cell. They have a cell wall present for protection. We've mentioned this. They are prokaryotic, meaning they have no membrane-bound organelles. We've spoken about this. They have a plasmid, okay, some rings of DNA or circular DNA. They have a number of different shapes. So first of all, if I get my pen here, they can be I'll fix that slightly. They can be rod-shaped, they can be spherical, they can be spiral, or they can be comma, okay? And finally, many of them do have a flagella, many do have a slime layer, and many have a capsule, but not any one bacteria looks identical to the other necessarily. So, the capsule, the pili, and the plasmid are probably three of the most important parts for us. Okay, so as the capsule, we've mentioned, just going over the same thing again and again and again, making sure you're familiar with it. This is a carbohydrate layer, and it is used for protection. Now, protection against what? Phagocytes, we're going to talk a little bit about them. Effectively, think of them as white blood cells within the body trying to protect our cells against these nasty bad bacteria. The pilus, okay, the pili, these are hairs that help the bacteria stick to a particular substrate, stick to a particular thing within your body. So they're going to try and bind to healthy cells and things like that. Finally, plasmid, this again is that small ring of DNA. Okay, it's only got a few genes, but it does provide some advantage to some bacteria against antibiotics, for instance. So, how do bacteria replicate? Number one, asexual, keyword. Okay, bacteria can reproduce by themselves in a process known as binary fission. This is very similar to mitosis as an overall process. Now, they can divide very, very quickly if they're given the ideal temperature, the ideal moisture, and enough food or enough energy in order to replicate. So how do bacteria cause disease? Well, we know that bacteria replicating, more of the bad thing within your body, and this is going to because they produce toxins, okay, and more bacteria, more production of those toxins, and they're going to cause either damage or interruption to cellular processes, or they might cause inflammation. Number two, they might induce an allergic reaction. They might trigger an allergic reaction within the body. Moving on. Our next topic, still within pathogens, is viruses. Okay. Now, you should be able to draw this very simply, same way that you should be able to draw bacteria very simply. Okay, you should be able to label some of these key components as well. So, number one, the outer membrane of this virus is known as the viral envelope. Okay, so some viruses are going to contain a lipid, so fat, or protein outer membrane. Okay, viral envelope. Now, in the center of a virus, they contain either DNA or RNA. It is never both, one or the other. Please include or in your responses. And finally, protecting that DNA or RNA within the virus itself is a protein capsule. So, commonly confused topic, are viruses living? And the answer is no. Okay, viruses are not considered living. And the reason here is that they cannot undergo cellular respiration. Okay? When we talk about what classifies a living thing, you might remember back to Mrs. Gren, so things like movement, respiration, sensitivity, growth. Viruses can do many things, but they cannot reproduce on their own. And so as a consequence, we say, are viruses long-living? The answer is no. So, viral replication is not as similar to bacterial replication as we'd like. It is a bit of its own process. So what are some of the key things to note? Well, number one, the virus itself binds to a host cell. What it does is it punches through the cell membrane and it injects its own DNA or its own RNA into that particular cell. The DNA from the virus then splices, aka it combines with the host DNA, and it acts as if it was a gene within that host's DNA. Now, this gene, as with many genes within the body, sends out instructions, and this instruction is to make this cell compose a new virus. So we've gone from our one virus up here, injecting its nuclear information into a cell, and now we've produced many viruses within this cell. We've started that replication process. However, unfortunately, the virus is going to cause this cell to produce so many virus molecules that eventually the viruses burst out from that cell and they'll travel and infect other cells. And this is going to lead to, in this case, where the cells burst, it being destroyed, or it could be significantly damaged as well. So, we've now looked at bacteria, we've looked at viruses, we've looked at some of the features and how they replicate. This is the key info that I would be focusing on for upcoming tests and exams.